Going to be recorded. Thanks for joining us for the second webinar in the eDNA project series. Today, our aquatic restoration and resiliency coordinator, Brittany Rogers, and our sustainable fish ecologist, Pippa Kahn, will give an overview of how eDNA will be used to detect the target species for this project. Next slide, Brittany. All right, and I just wanted to take a moment to introduce myself, and then my colleagues are going to do the same. I'm Megan Pistoles. I'm the Education and Outreach Coordinator with SLELO PRISM.
Pardon the interruption. Are you showing slides?
Should be showing slides, Brittany. I see slides. Does everybody see slides?
Yes, I do.
Must be at my end. I do not see anything.
I see them.
Interesting. Okay, well, maybe we could pause for a moment. Brittany, do you think there's anything going on here that we can fix? You must have to hit your view, Rob.
Yeah, I would say check your view. It sounds like it's a computer.
Okay, so moving on. Brittany, would you like to introduce yourself?
Sure. Thank you, Megan. So I'm Brittany Rogers. I know most of you on the line, and thank you so much for joining us today. I'm the Aquatic Restoration and Resiliency Coordinator. So I primarily focus on aquatic invasive species, including field surveys and different special projects that we do, including this eDNA project and some restoration work and survey work out in the field. So thank you so much, and you'll hear more from me in a little bit.
Pippa?
Hi, I'm Pippa Kahn. I'm in Rochester, New York, and I work for the Nature Conservancy on our Great Lakes Sustainable Fisheries team. So you'll be hearing more from me and more specifically about the work that I do on Coregonids in a few slides. So thanks for attending today.
And since we have them on the line here, how about the SLELO PRISM Program Manager, Rob Williams, and our Volunteer Programs Manager, Mary Ripka, could say hello.
Hi, I'm Mary Ripka. I'm the New York State Volunteer Programs Manager. And my actually primary focus is the volunteer programs, but I also do administer a hunting program. And I just want to let you know that you may know this, but I didn't catch that fish in Lake Ontario. But it was the best experience of my fishing life. So I will say that. So I kissed him and I sent him back to the ocean. So.
Hey everyone, Rob Williams here. I'm using dual monitors and I finally figured out after all this time how to make them work. So yeah, as Megan mentioned, I'm the manager of the SLELO PRISM. Been doing this for about 10 years now. And I just love learning more and more about eDNA. We started a program, I don't know, 5 years ago or so. Did a lot of very interesting work, learned a lot about eDNA. And we had finally incorporated it into a core piece of our programming here in terms of early detection. So really interesting stuff and very beneficial stuff, not only to native species, but invasive as well. So kudos to the project team on this one.
Excellent. Brittany and Pippa, would you like to take it away?
Sure thing.
And
thanks, Megan.
I think we're going to turn off all of our video. Is that right? Yeah. If everybody could kindly make sure you're on mute and stop your video, we would appreciate it. Thank you.
Great. So Megan kind of touched on this, but just to give an outline for what we will be going over in today's webinar, I'm going to do a quick recap of what the project is, just in case you missed our previous webinar. And then we're going to dive into the world of eDNA. So what is it? How do we use it? How are we planning to use it for our project? And I will keep this at a higher level. I'll put the preface out there that I'm a fish geek, but not a lab geneticist. So we'll get the basics that we need to know to plow forward. And then we're going to dive into our target species. So the native as well as the invasive species, everything that would be good to know about them, so you can get an idea for what it is that we're looking for in our sampling this year. Next slide, please. So as a recap, we got funding to really expand the eDNA work that has been occurring through SLELO. And so we are looking at collecting eDNA samples from down really near Oswego in the south up the eastern lakeshore, and then into the St. Lawrence up to Messina, New York. So we're going to be looking at tributaries from August through December. And so really there's two purposes. The first is to see if we can detect presence of Coregonids. So this is like whitefish and cisco during their spawning season, which typically occurs in November and December. And then also look for the presence of invasive species in these same habitats. And so by sampling from August to December, we'll kind of pick up on the tail end of the summer and then into that spawning season and hopefully increase any detection probabilities. Next slide, please. So eDNA, buckle up. All right, next slide. So your first question might be like, what is eDNA? So first off, what it stands for is environmental DNA. So this is DNA that can be found in soil, it can be found in water. And so we have this little picture of this fish right here to kind of show you the various ways that DNA can actually enter, for our case, into water. So, you know, it could be a dead fish, so cells could be coming off of that. Fish might lose scales. This is quite common, and those scales will then kind of float in the water and down to the sediment. Feces and mucus, also for those that spawn and deposit eggs, those materials will also be giving off cells. So what we really want is down in the water is to filter out the cells, because that's what we are needing for our laboratory analysis. But it's quite interesting when you look into the myriad of ways that people have figured out how to collect DNA. So again, our focus is aquatic, but if you're at all interested, there's a lot of interesting literature out there. Next slide. So one step back, so you might remember from like 7th grade biology, DNA, deoxyribonucleic acid. We have two forms that are typically used in these types of studies. So the first is nuclear. So this comes then from the nucleus in the cell. And you'll see the chromosome and how it breaks down and how we have the base pairs, adenine, thymine, guanine, cytosine. But we can also find DNA in the mitochondria in the cells. And so those little pictures there are representative of our mitochondrial DNA. Typically, the mitochondrial DNA is used because the sequences are a bit shorter. They've been studied for various species. And as you'll see in the cell, there's actually like numerous mitochondria. So you can really get a lot of the copies of the DNA, which helps with our analyses. Next slide, please. So I'm going to focus really kind of on the aquatic side here on some of the uses. But first is just simple species detection. So is something present in the water or not? And as a fisheries biologist, this is really exciting because sometimes to look for species, it takes a lot of field work. You have to invest a lot. So the fact that this can help kind of streamline some sampling is really exciting. But also when we're thinking about working with any rare species, whether they're listed as endangered or threatened, what's really nice about collecting eDNA is that you're not killing a fish to have to get a sample. You're not stressing them out. So it offers a really nice way to collect needed data without causing any harm to the species. It also helps with these cryptic species, so those that are like well hidden, or maybe even those that don't respond to sampling efforts. So a lot of sampling gears often have biases and they might not effectively sample all species. So that's a really nice thing about eDNA. If there's cells that are deposited and you can collect them, then you can look for presence. They're also used, we also use a lot of eDNA surveys to help with understanding biodiversity. So we can look at community composition. So if we collect some samples, we can see what different species are there, the assemblages, who makes up that community. And again, it's a really efficient way to sample across various species. And then as I'll be talking about in like a slide or two, it also helps us get into behavior. So for fish, we're often thinking about where they're moving to, where are they spawning. So again, by being able to collect a sample, we can see if they're present. We can look at this over time, and it really helps us provide another tool in the toolbox for really addressing a lot of these important questions that we think about as fisheries biologists. Next slide, please. So in order to do our eDNA sampling, we've broken it down into a lot of the basic steps, if you will, of what we'd go through. So the first is actually just going out in the field to collect the water samples. At the bottom left, there is a picture of Brittany grabbing some water. So once we grab that water, the next thing that we have to do is actually concentrate the DNA. So we do that, there's really two ways. One, you can centrifuge it. So if you remember, centrifuge just spins things really fast and separates out. Or you can use a pump. So you basically pull the water through the pump and everything ends up on the filter, which is what you're seeing in that second photo there with like the little tweezers. Then once we have this kind of concentrated material, the next thing is that you actually extract the DNA. So use some chemicals to help with this, because then you'll take that sample and then we'll go to the lab. And then the next thing you're looking to do is to what we call amplify. So it's really looking to increase the amount of DNA that's there, and so it can help us actually detect what's in the water sample that we're looking at. So the bottom right is really the end output on what we call an amplification plot. So it kind of gives us an idea of where that red line, the threshold for detection, and anything above it, if we know the DNA going in, it will tell us if it's above it and if our sample has indeed detected that. So really efficient, really cool process. Next slide, please. So just to dive in a little bit deeper. So with eDNA, we have two really cool techniques available. So the first is what we call a qPCR. It's a quantitative polymerase chain reaction. So essentially what you're doing is you have a set of species that you're looking for, and then you test for those species using primers, which I'll talk about in a second. And then the other method is our next generation or high throughput sequencing. Essentially, you haven't selected any species in particular. Instead, you have more of what we consider like a library of genetics for various species, and you run your sample and basically see what pings. And so this is often like an approach that's used to help understand like community composition. It's really kind of still a burgeoning field, if you will. So with our work with qPCR, you will need primers. So for example, let's say we were studying largemouth bass. So we would have a little DNA code of largemouth bass, and then we'd use that to actually see if our water sample contains that same DNA. As you'll see there, exemplified in like the red, the green, the blue, and the black, this is representing that adenine, cytosine, the various parts of the DNA. And they're kind of like puzzle pieces in that certain pairs go together. So if we have the same grouping, we should actually see then that DNA get replicated in our samples and tell us that the DNA from that target organism is there. However, if we have that primer and let's say there was no largemouth bass in our little fake scenario here, then we wouldn't have any matches and we wouldn't have anything being copied. So we wouldn't be able to detect largemouth bass. So that's it kind of at it. It's very simplest. So I like this image quite a bit. Next slide, please. So these are just some photos from the lab, because often we're field biologists. So this is kind of the other side. So in the upper left, that's actually preparing the trays, which are then used in the bottom left, the machine that will help actually run all these qPCRs. And in the right is just a photo of what it looks like through the software when it's producing those amplification plots. Next slide. So for this project in particular, we are just focusing on species presence and absence. So we'll have a suite of native species as well as a suite of invasive species that we'll have primers specifically developed for that then we will be testing for. So we will be collecting water samples at those sites that I showed you in like the third or fourth slide. Typically you gather about 1 liter, and we'll also be doing the vacuum pump method for really concentrating the DNA and extracting it for lab analysis. And we're going to be utilizing the qPCR method as we have our suite here, and we're hoping, as you'll see on the right, to get an amplification plot like this, which is showing in this case the presence of goby in the sample. So fingers crossed we get this for some of our cool species, native wise, that we're looking for, and maybe not for our invasive species. Next slide. So now we're going to actually talk to you guys about what are we actually looking for out there. So we're going to start with the native species first. So, you know, as much as we'd love to really sample for everything, that wasn't realistic at the moment. So in order to be included on our testing panel, there were several things that we thought about. The first is I do work on our Great Lakes Sustainable Fisheries team. So it really had to fit in with our strategy, which was a large part of what helped form the native side of this. So I work with Coregonids that I'll talk about in the next slide. So of course, we'll be looking for those. But then when we're thinking about other native species, several things came to mind, like what else might be using those same habitats so we could potentially detect them as well. What is their status in Lake Ontario? Are they really abundant, or are they quite rare? You know, if it's something that's really abundant, you know, what would this study be telling us versus if it's kind of rare, maybe we could be helping out. Also considering their role in the food web. And then TNC likes to partner on lots of things. So we didn't want to be doing this work in a silo. So if there was any potential for collaboration and helping the greater knowledge on understanding where our native species are, we were hoping to help with that. Next slide. So this is a really simple summary of our Lake Ontario food web. So at the very bottom in the green, we have our primary producers. And then we go up the various stages to the very top there in purple, which are our top predators called piscivores, our fish eaters. So if you'll hit next, Brittany, this will highlight some of the ones I'm going to be talking about today. So on the very far left is lake whitefish. So you'll see that they're in really what we call like the kind of the forage fish category. So they're often prey for things like our rainbow trout or walleye or lake trout, those piscivores. And then we're also going to be talking about the American eel and the Atlantic salmon, which are piscivores as well. Just the way it was put together, they got kind of grouped lower down. Next slide. So you'll hear me say Coregonids, Coregonids. So really what Coregonids are, they're a subfamily of the salmon family. And it's really interesting. I might just say that because I'm a fish person, but there's quite a number of different Coregonid species out there. So you'll find them in the Laurentian Great Lakes, up into Canada, in our inland lakes, and they're also present in Europe as well. So Coregonids can be deep water, so you may have heard of bloater or kai, and they can also be found in the near shore, so like our lake whitefish and our cisco. So today and for our sampling, we'll be focusing on lake whitefish and cisco. As a team, this is our interest area because these fish have served important roles in the food web as that forage, that prey for top predators. And in doing so, they typically help to stabilize the food webs. But what's happened is that we've seen in a lot of the lakes, their populations have declined. And so what happens is we have our invasive species come in and we have these less resilient food webs. And so we'd like to see them kind of rebuild and recover. So by understanding with this project where they're spawning, that is, you know, another important thing to know on their recovery. So if you'll hit next. So as I mentioned, they have faced, you know, quite a number of declines. This is what you're seeing is lake whitefish on the top and cisco on the bottom. And this is the commercial harvest in Canada and the US just in Lake Ontario. And while commercial catches aren't always the best indicator of abundance, this does give you an idea of how they were once, you know, quite abundant and, or at least in the commercial catches and now aren't. So several reasons for this are simply, you know, they were fished pretty hard. You know, these are quite popular fish back in the day in industrial times, and there was a lot of fishing pressure put on them. We also saw the introduction of invasive species, and like specifically the alewife had a real big impact on their populations and really kind of affected their status in the lake. And then we've also had, you know, changes in our climate and the environmental conditions within the lake as well. So they're under a suite of pressures, if you will. Next slide, please. So something that we're seeing is that historically, some of these populations of lake whitefish in our upper Great Lakes, like Lake Michigan, they used to use tributaries to spawn. And we're very interested in understanding where these fish are spawning because that's kind of the puzzle piece to try to help and rebuild their populations. And recently, the very top is a picture of a journal article from 2021. We've actually seen returns of river running lake whitefish in Green Bay, which is really exciting. And in Lake Ontario, this question of whether they might be using tributaries is really unanswered. So we don't really know. And so that's what we're really hoping to get to with this project is, are they present in these tributaries during the spawning season? Because this is the first step in understanding whether this might be a strategy that we're using that has not previously been thoroughly explored. Next slide, please. So another native fish that we're going to include in our testing panel is Atlantic salmon. So like Coregonids, they were once abundant in Lake Ontario and they were found in the St. Lawrence as well. You'll find them in the Northeast of the United States over into Europe. If you ever go to an event and have salmon, you're probably eating aquacultured Atlantic salmon. So, you know, they're around, but unfortunately in New York by about the 1900s, they were gone from the lake. Which is quite sad because they once supported a large fishery. A lot of the settlers into the United States really depended on the fish as well as the native people. So they're an important staple in Lake Ontario, but they faced several pressures, which I'll talk about shortly. They have a unique life history. I'm sure many of you know about salmon. So they live in the ocean and then they move into freshwater to spawn. So they're using some of these same, you know, or potentially using some of these same tributaries as where we'll be looking as Coregonids. They move in about the fall and will spawn. And what's really neat in Lake Ontario is that some recent work has shown that there is actually a landlocked population of Atlantic salmon. So instead of doing their traditional movements all the way through the St. Lawrence out to the ocean and then to come back in to spawn, they actually stayed within the lake and its associated tributaries. But you know, like many things, they were they were taken out. But that's a really cool finding that came out recently. Next slide, please. So you might be wondering, okay, what caused a lot of the declines in our Atlantic salmon? So the first is just simple presence of barriers. So as you might recall, the Northeast has a plethora of dams that were built, and for migrating fish species, this is never good. It's hard to maneuver around these. They have involved salmon ladders or fish ladders to help move along the sides and get up and over, but it still had huge impacts on this fish. Another one is pollution. So with that industrialization, we saw changes in agriculture practices, deforestation. And so there was a lot of like sediment and material entering these tributaries and these streams, and it really degraded the water quality. As I mentioned, there was huge fisheries. So unfortunately, as is typical with a lot of fisheries around the industrialization period, we saw overfishing of these populations. And then we also have, again, the presence and introduction of aquatic invasive species, which can impact food webs, habitats, you name it. Next slide, please. And then the other native species that we're going to be including on our testing panel is American eel. I personally find the eel so interesting. You'll see on the right here a map of their distribution. Eels are found globally, and for the American eel, you'll see that they can be found up in Canada, down through the Northeast to the South in the Midwest states, I mean, down through Mexico and the Yucatan Peninsula, the Caribbean, Central and South America. So they were widespread. They could really survive in a diverse range of habitats. And I even read one fact that said historically, eels were about 25 to 50% of the fish biomass in streams. So they were really abundant. But unfortunately, like our Atlantic salmon and like our Coregonids, their populations have also faced declines. And it said, I believe for the Moses Saunders Dam, I have the fact here, there was a 99% decline in juveniles ascending the Moses Saunders ladders to Lake Ontario between the period of 1983 to the mid-1990s. So we've seen almost a threefold decline in the populations in our waters. Next slide, please. So eels are really interesting, and they have almost the flipped life history of the salmon. So they're catadromous. So they live in freshwater and then they'll actually move to the oceans to spawn. We don't know much about their spawning, but do think that they spawn in their Sargasso Sea. And so they'll spawn, they'll have eggs, they'll turn into this really cool larval stage called a leptocephalus that kind of looks like a clear leaf. And then they'll turn into glass eels. And at this stage is when they'll start moving into freshwater. So from when they're glass eels into elvers into yellow eels, they will move throughout the system. So, you know, think about going from their Sargasso Sea up the St. Lawrence and into Lake Ontario. It can actually take several years. So it's not an overnight thing. So when you think about pressures on these species, not only do they face pressures in the marine environment, but then they face pressures in brackish and then the freshwater as well. So next slide. So not surprisingly, there's also many challenges facing American eels. Like salmon, the introduction of all these barriers to a migrating fish species is quite difficult. Again, they have put in fish ladders, but when you have such extensive migration patterns, that can be difficult and affects several things, like even like water properties. So that has had an impact. And also with hydroelectric, some of the larval and juvenile stages will actually get caught in there. So that has had a huge impact on this species. The middle photo is a photo of some of the glass eels. They're really cool. But unfortunately, when I talk about overfishing on these populations, that's something that's happened historically. And it's also something that is happening currently. So in a lot of places, you will find that when you say fishing for eel, it's not just adults. So the glass eel fishery is a fishery in and of itself. And actually it's illegal in the US. And so this is a photo from the National Geographic magazine detailing some poachers that got caught in Maine that were trying to sell them for $3,000 a pound. So for an at-risk and rare species being hit by fishing at your early life stages as well as adults, it's had a huge impact on the population. And then of course we have changes in our habitats as well. So a lot of those same things that are degrading habitat quality are affecting the eels as well as the salmon. I just threw in the picture to the right. So in the Northeast, if you ever see these kind of rock piles in a V shape, they might be remnants of an old eel weir. And that's what this is. So there's, really small fishing efforts on them, kind of, Joe Schmo out there with his eel weir to large commercial efforts. So yeah, the eels are quite interesting. They're not listed in the United States, but in Canada, in Ontario, they're listed as endangered under their Endangered Species Act. And then nationally in Canada under the Species at Risk Act, SARA, they are listed as threatened, I believe. And then they're also listed as endangered by IUCN. So again, another species that if we could detect, it'd be really great to know, and we could contribute to our greater understanding of how our native species are faring in the Great Lakes.
Thank you for the wonderful introduction to native species, Pippa. So now we're going to transition over to some of the invasive species that we plan to include in our samples. I will say there's a caveat to that. So as part of what Pippa mentioned earlier, developing the primers is something that is kind of seems very straightforward and simple, but if you are a trained geneticist, you know that it is a little bit more difficult than that. So we are still working through some of the primers for our invasives and native species, and hopefully all of these will be included in our results. So as Pippa mentioned, with the native species, we couldn't include every single thing that existed out there, and we included as much as we could. And one of the ways that we did this was based on the SLELO PRISM programming focus and also collaborations. We also really needed to consider the negative impacts of certain invasive species to those native fisheries that we talked about earlier. And then also some of the species that we chose, we included them partially because we also just don't have a good understanding of their distributions in the SLELO region or in the Great Lakes in general. And this project can also aid us in future management. So the first species I'm going to talk about is our Asian carp group. And so these are fish species that are native to Asia, and there's 4 of them: the black carp, silver carp, bighead carp, and grass carp. And I will mostly focus on the black, bighead, and silver when I'm talking today, because there are some cases where sterile grass carp are permitted to be released in certain water bodies to control algae. So the Asian carp group are voracious eaters, and they consume plankton like algae and other microscopic organisms. So thinking back to that food web that Pippa mentioned, that whole bottom line there, these species, that is their food source, and they eat a lot of food. And so having that, and then also the fact that some of the females of these fish species can lay up to 500,000 eggs each time they spawn, and they have no natural predators. So they're laying up to 500,000 eggs, and they're eating a lot. So as you can see, their populations can really increase quickly. So I'm just going to quickly mention the ID of some of these species. So the black carp is the darkest in color of the carp group. There's no scales on their head. They have a pointy face. And here is a picture of the actual fish. And one of the weird things about the black carp too is that they have molars in their mouth that almost resemble like human molars. So then we have the silver carp, which is very silver. This is the species that you see those videos or those crazy images of thousands of fish jumping out of the water in the Midwest where they're located. These species don't have scales on their head. They have a very like frowny face or a downward slanting mouth and very low set eyes. So as you can see, that frowning face and low set eyes, very silver fish. The next species is the bighead carp. And so this species is a little bit darker in coloration than the silver. And again, there's no scales on their head, no barbels in their nose, that sad frowning face. And one of the things, I'll pull up the picture of it, is they have a really low set eyes. So pretty distinguishing things with the with the carp group. And then the same thing with the grass carp, which is more common. And this is one of our larger fish species that some people might be familiar with. So just quickly want to mention the distribution of some of these species. So the black carp is located in the Midwest up the Mississippi River. The silver carp is a little bit more widespread and has actually gone into a lot of other tributaries and waters and has been identified in separate areas. Although not in the Great Lakes, as far as we know, there are some samples that have been collected in near Chicago in Lake Michigan. And then the big head carp is a little bit more widespread through reports. And again, it's not actually known to exist, especially in Lake Ontario. So those are some important species that we definitely want to test for and at least figure out if there is any kind of DNA present in our waters. So the next species is the swamp eel. And so this might be new to many of you on the line who have worked with SLELO before. We haven't really talked much about this. The swamp eels are identified by their absence of pectoral fins. They don't have any limbs at all. And they have like a single V-shaped gill opening. So that is the main ways that you can identify them. And there's concerns with this swamp eel because they can displace our native species because they eat fish, shrimp, crayfish, frogs, eggs, and other aquatic invertebrates. So impacting that food web pretty strong. And then also they have really intricate and extensive burrowing systems. And so there's concerns of the acceleration of drying shallow water bodies because of those burrows. So one thing I want to mention, so we talked about primers. So this is one species that we would just be going for that Monopterus species, the genus group, and it would not be species specific to the albus or the cuchilla. So we would just be testing for the presence because any swamp eel presence would result in further assessment required. So the swamp eels are morphologically similar to some of our North American species like the American eel or the invasive sea lamprey, but also there's 2 native salamanders, which you can see on the screen here as well. And just remembering that absence of pectoral fins, absence of limbs, and V-shaped gill opening, then you would be totally fine to distinguish this species from others. This picture was actually taken in Hemlock Lake in the Finger Lakes area or like towards Western New York. And there were 12 adult swamp eels that were found there from a student that was going to school out there, and he was out fishing, and the adult species had washed up on shore. This was identified as the cuchilla species, and this is actually known to exist more in like rice fields or mud fields out in the like Indian subcontinent. So just kind of thinking of that, the state has decided or announced that they believe that this species was actually the 12 adults were actually released as adults, and that there's not a population that has been established, although they're continuing to assess the waters in Hemlock Lake and try to find if there has been any of these species established. I think this species is a really good indicator for us to first test for their existence, but then also to continue to emphasize on the need to detect species really early and be able to have a response prepared, and also to educate people on how we can protect our waters from new species introductions by not releasing species into our waters where they don't exist. Our next species, I'm not gonna mention the distribution just because it's not truly known to be in New York or established in New York State at this time. So there is no distribution. There are some areas in Pennsylvania though for the swamp eel that it is believed to be established. The next one is the northern snakehead. This is a fish species of much concern to many people. The ways to identify it is with the single dorsal fin that runs down the entire length of its body. You can see in the top image there, or on the bottom, you can kind of see it, but the dorsal fin is down in that image. And the top image you can see is actually a juvenile, and the one below is an adult. There are some studies that can also distinguish between males and females by the coloration. So they have like that light tannish coloration and the dark blotching, but they say that males get a lot darker in color than females. Again, these species are impacting our natives as voracious feeders and also competing for habitat. They're very aggressive fish, so they eat zooplankton as juveniles and like smaller larvae or crustaceans. But then as they grow into adults, they eat almost exclusively other fish, which is 97% of their diet. And you can get a really large snakehead, as you can see in the bottom image there, and they can actually eat a single fish that is more than 30% the length of its body. So if you can imagine having that for dinner, that is a huge meal, and they continuously do that throughout their lives. So big impact on our food web. I do want to mention they have some crazy teeth in their mouth. I just love this photo of the snakehead, but there are two native lookalikes, the native bowfin or Mia calva and the native burbot or Loda loda. So with this species, the best ways to distinguish them between is the position of the pelvic fin. So directly, if you look at the snakehead's pelvic fin, it is directly behind the pectoral fins. But if you look at the bowfin, it's further back. And then also the bowfin has that really dark eye spot on its tail. So you can see that for the males or the juveniles, but not all bowfins will have that you can see. And then of course the burbot has that more similar like blotchy or more camouflaged flesh that you can see, but there are ways to distinguish with the barbels on the burbot. So just a couple ways, and then also the dorsal fin as well. So keep a lookout if you see snakehead or you know someone that thinks they saw snakehead, get photos of the face and mouth, and then of course that dorsal fin is really important as well. The current known distribution of this species is in New York State in the Delaware River. There's been quite a few found there, and then also down in the Long Island and Lower Hudson region, there's some ponds that northern snakehead is known to exist. Talking about the next species is tench or tinka tinka. I just love their Latin name, tinka tinka. So these are native to Europe and Western Asia. They're actually a minnow of or a member of the carp family, and they can grow up to 18 inches in length. And you can see that they have this dark olive or like almost like golden colored body with dark fins and tails. And one of the crazy things about these guys is they have a really deep red eye. So that's a good way to identify them. And then also they have the terminal mouth with the barbel in each corner of their mouth. I assume most of the time if somebody is out fishing, they would know if they caught a tench because there are no other species that I think look similar. One of the things with tench is that they do prefer very weedy and muddy water bottoms. So they have high reproductive rates as well as long lifespans, and they can survive in low oxygen environments. And just to kind of further connect the world of invasive species, thinking about aquatic invasive plants, they grow into large dense mats, and when they start to die off and decompose, they actually release large amounts of phosphate, and that actually lowers oxygen levels, so becoming an ideal habitat for tench. But then also that large abundance of phosphate they're releasing has been connected to harmful algal blooms. So kind of thinking of that like triple whammy there, you have invasive plants, tench, and potential harmful algal blooms all in the same areas. So just kind of thinking about that, it's extremely important for us to understand their distribution, because currently there's some known reports in Lake Champlain and in the St. Lawrence, but you can see the only reported tench is in Cornwall, but we do know and we have heard of reports further out in the St. Lawrence River. So we're hoping to at least use this study to get a better understanding of potential distribution and then further target surveys moving forward. So talking about the tube-nosed goby, I think one of my images is missing, so I apologize for the big blank white space for you all there. So this is a lesser known or discussed species of the goby family. Many people are familiar with the round goby. It's very common in a lot of the regions, especially in SLELO. This is slightly different and can be identified by the 5 dark bars that they have on their sides, and then the scallop-shaped pelvic fin underneath that kind of creates like a suction cup. So that's similar to the round goby. But then if you look at the tube-nosed goby picture on the bottom, you can see their nostrils are actually tube-like and stick out from their face a little bit. So easy way to identify the tube-nosed goby. And our current understanding of their distribution is missing right now. So we are hoping to catch these guys because they have the same preferred habitats as many of our native fisheries and nurseries. And the males of the tube-nosed goby are especially aggressive, and they guard nesting sites and will defend their eggs and their young. And then the females can actually spawn multiple times a year. So just think about those populations. Just a quick comparison, the round goby and tube nose goby, the top two fish you see here, are both invasive. The round goby has the black spot on, you can see in the top image there. And then one of the major differences is we do have the slimy sculpin, which is a native species. And so thinking about this, there's no spot on its dorsal fin. It doesn't have that suction cup underneath like both of the goby species, and they don't have scales. So they are slimy, which is why some people call them slimy sculpin. So just kind of thinking about that, if you see any fish out there that resemble the gobies, or the sculpin, you can think more about that. So as of right now, there are some people that have research and information on the distribution of the tube nose goby, and we don't currently have that. So as of right now, there's only a couple reports in the upper or the like mid St. Lawrence region, in our region, up by like Grass River and Messina area. However, so I mentioned there is some research. There are some professionals out there that are trying to study the tube nose goby. So we just want to help get a better understanding of their distribution. And then the rusty crayfish is another species that might be underreported in our region. We've recently heard more information about its existence in the St. Lawrence River. And so the issue with the rusty crayfish is they are more aggressive than our native crayfish. So they're displacing our native species, and they're less likely to be preyed on by fish just because of their aggressiveness. And they reduce the shelter for young fish and other aquatic invertebrates, and they also can prey on eggs and other species. One of the interesting things with the rusty crayfish, though, is they are actually native to the Ohio River Basin, so they're not really that far from New York. However, they were introduced as a bait species, and so they really are not native to Lake Ontario or any of our inland tributaries or waters in our region. So you can see it has a pretty wide distribution in the like capital region from Albany down around the Catskills and starting into the Finger Lakes. The pink dots you see on the screen here are reports that have not been able to be confirmed. So that is something to take into consideration. And then we recently got a report up in northern New York. So there's a huge region and a huge gap between where we know they exist and where we think they might be. So again, hoping to use this project to help us with that. So I'm going to pause for a moment and kind of transition away from the species. That is the end of the species that we are hoping to include in this project. And I just want to talk about DNA again a little bit, just to kind of bring everyone back and kind of hopefully bring everyone onto the same page. So I just want to mention that our 2021 eDNA testing is to solely look for the presence or absence of species and not really looking at population size, what they're doing in the waters. The results of this study will result in follow-up actions from our teams and, maybe potential future studies in the coming years. But this is not to say 100% for certain that species are in existence and established in these waters. Thinking back to what Pippa had mentioned, that there's many different ways that DNA can end up in our waters, and it does not always mean that that species is established or dwelling in that area. And so I just want to talk about some of the different impacts on eDNA and how they might impact the way that we perceive our results. So starting with the picture on the top left here, so the origin. So the origin of DNA, Pippa had that one picture of the goby with the different ways that you can get DNA from it. And so just because we have that presence or absence doesn't necessarily mean that that species is alive or in existence there. So there was one study that showed that juvenile sunfish actually released their eDNA at a higher rate than adults. And the study actually found that behavior and metabolism might influence the eDNA production. So younger fish might increase your chance of collecting a presence. And then there's also been studies where they're actually testing the fecal matter and analyzing fecal matter, not to determine what species released that fecal, but what species that, or what kind of animals that species was eating. So finding DNA in results from fecal matter, like birds, is one example that we can use. So if the bird traveled from somewhere else where they ate a fish, that might be giving us a false presence, so to speak. So what we're doing is our efforts are going to maximize our success. We're hoping to maximize our success by taking into account temporal or seasonal events like mating. So thinking back to the Coregonids, so they should be spawning when we're out there sampling, but then also like spatial patterns. So the migrations or known habitats, and we're targeting our sampling windows and areas to try to increase our success at detecting a more true presence. So then moving to that image on the right, this is just a little bit more of the lab side of things, but we actually, you saw that picture of the filter. And so the different filter sizes can also give us different eDNA results. So just because we have a huge amount of DNA from a filter does not necessarily mean that there's a huge amount of fish in that region. It could be that there is high amounts of decaying material, or that the particle sizes were larger and captured more easily. Moving down to the transport aspect on the bottom right. So it's important that we're including this as part of our consideration and thinking about where we're studying in tributaries. So DNA is spread and transported throughout water. Unfortunately, water is very fluid. So just because something is left somewhere doesn't mean that's where it will stay. So we have to really think critically about the species presence in space. So was the species super close to where we actually collected the water sample, and how long or how recently was that species in that water body for, because a fish could swim up a stream and back out and we can still detect a presence for that species. So thinking of that, and then also the sediment in water bodies actually can hold DNA. And so if that is stirred up, it can actually release old DNA that has remained in the system for some time. That could also give us false presences. So just thinking about how our results will guide us, it's really important to remember that this eDNA analysis is only the first step in our studies, and that we have a lot more to do. Moving to the left side, the fate. Really, this is just talking about how long the DNA exists in the system before it decays. And there's been a ton of studies out there based on environmental conditions of temperature, flow, the population, the size of the species, things like that. And that all plays a role in the detection or the presence of or an absence of a species. So kind of thinking of that, there was one study in a controlled environment where they had put a New Zealand mud snail, which is an invasive snail, into a freshwater system, and that DNA remained in that system for 21 days after that species had been removed from there. So 21 days afterwards. So just thinking about that, we will respond to these results, but just because there's a presence does not mean necessarily that that species is in existence. So I think there are caveats to eDNA, and I think it's really important to talk about this, and also to get everyone on the line and everyone else thinking about this with us and considering some of these ideas, and that we are doing this as a first step. So we're testing in or near the mouth of the tributaries rather than doing transect sampling where we're collecting dozens in each water body, because we just want a sample of each water body, as many tributaries as possible, so that we can get a better look at the overall scale of things. And so for anyone else on the line who works in aquatic environments, how many challenges there are for working in the water, but aquatic ecosystems really truly are critical components to our global environment. So in addition to them being essential to biodiversity and ecological productivity, they also provide services for humans, like water for drinking, irrigation for farming, recreational opportunities, and then also habitat for economically important fisheries like the Coregonids and salmon that Pippa had mentioned earlier. Unfortunately, our aquatic ecosystems are continuously being threatened, both indirectly and directly by humans. So it's really important for us to take this step to get a better understanding of what exists. But if anyone knows who has gone swimming or been around water, it's really difficult to see everything that's under the water. So thinking back to using this as the application of environmental DNA as an effective surveillance method in our toolbox, it's just one tool that we have. And, we'll continue moving forward using different tools after this, but this is just the first step. And we will be able to, hopefully in a less intrusive way, detect populations or low detections of both the natives and the invasives, and increase our ability to respond to either contain the spread of the invasives or protect the natives that are spawning in these tributaries. It's really cool because with eDNA collections, you're just collecting a sample of water out there, and you're getting results are like sight unseen. I saw written somewhere the sight unseen, so you're getting a presence or absence record of something that you never actually saw. These advancing technologies have really been changing over time. The cost has been decreasing and making eDNA a higher potential for detecting species. So it's really important for us to be able to use this. It's more cost effective than traditional surveys, and it will also help us really target our focuses moving forward. So whether we're out there collecting eDNA for the presence of low abundance populations like our Coregonids in these tributaries, or trying to detect invasive species, we are using this innovative tool that is continuously proving to be essential in the protection of our waters. So thank you for your interest, and whether we're out there looking at invasive species under the water or native species, we hope that you guys will all join us out there. So now I'm going to quickly mute myself for Mary to talk about volunteers for this project quickly.
Yes, thank you, Brittany, Megan, and Pippa. All that was amazing information. So yeah, so where we need your help actually is, so the SLELO team has ordered a mobile lab that will need help setting up, which includes unpacking and supplies, setup of the lab itself. We've also need collecting water samples. So once those are collected from the various pointed areas, we'll also need help transporting those water samples to the lab, which may be Oswego State. We're not 100% sure on that, but so we'll need help with that as well. And that would consist on driving them directly out to the lab. Collecting the water samples, just a quick backup there. That would be some on the shore, some from boat. So just make sure that you know that. And then capturing photos and videos, that would be huge for the end report and for information as we're going along as well. So if there's anything in there, you can volunteer for all of them. You can volunteer for just one. You could volunteer to be our photographer. It's totally up to you. And once we get into the portion of, where you are volunteering, then I can actually I'll be available for you for any kind of information that you need, any help that you need. I'll be there to assist in any way that I can. So thank you all. Thank you very much for joining us and helping us out.
Thank you so much, Mary. And so we're reaching the conclusion of our PowerPoint. Pippa, I don't know if it's okay with you if I just continue to close out. I'll keep going unless you stop me. So our next webinar following that is just actually the 411 of being an eDNA volunteer. So we did choose to have this webinar in the evening, and our purpose was that because we're hoping to better engage some of the volunteers that want to participate in our program and assist us. So you'll be able to ask more in-depth questions about the processes of what we're doing. We'll share more direct information about what we're working on. And then also Mary will get you set up with all your forms and everything else that you're working on. You can register for that on our website, SLELOinvasives.org/eDNA. And Megan is going to release a poll. I don't know. Perfect. Right. There we go. So if anyone online is interested in volunteering or assisting us with the collection of water samples, the mobile lab setup, transportation of samples, taking photos or videos, anything like that, please let us know. I'll just give one more second. Excellent. So thank you all who answered the polling question. I'm going to end that now. Just move that over. And Megan, I'll share the poll results with you following this. And So I guess at this point, I would like to say thank you to our funders again, the Orconect Foundation, for funding this project and making everything feasible that we talked about today and in our last webinar. So I'm going to go ahead and stop talking now. You guys have heard enough from me today, but if anyone has questions, please send them in the chat or feel free to unmute yourself and we can discuss. So thank you so much again for joining, and I hope to see you out on the water this summer.
Well, it's a great presentation. I learned a lot. Thank you very much for discussing all this information.
Yeah, thank you so much, Megan. So Frank asked when sampling starts. So we are hoping to start in the end of August, and we will continue sampling through mid-December, right, Pippa? I think early mid-December, we're hoping. Yeah.
Is there like a map already laid out of areas you're looking to sample?
Yeah, so we're kind of working through this juggling act right now. Let me just pull this up really quick for you. So we're working through this juggling act, and basically you can see the stars here along the eastern Lake Ontario shoreline and all the way up through the St. Lawrence. Those are the tributaries that we're targeting, but all of this connects together with the cost of the samples, the cost of the trailer, and all of our budgeting. So Pip and I are juggling all these things still as we're trying to bring everything into play. So we'll know by the August date our locations.
And how far up the tributaries are you looking to go? You're looking just at the mouth where it enters the St. Lawrence, or going back up to some of the headwaters or main branches?
So we would be a little bit up in the each of the tributaries to ensure that we're not collecting DNA that's existing in the lake, but that's something that came from the actual tributaries. And we're also targeting habitat locations. So Pippa can speak on that more, but we're actually targeting specific areas that we can see or we would believe that fish would be spawning. So those are ideal locations.
Yeah, and for the Coregonids, what they're finding in Green Bay was that on some of the rivers, a lot of the habitat used by walleye is actually indicative of habitats that the Coregonids were using. So by sites like the Oswego and the Oswegatchie, where there is a dam, down from the dam in like the riffle habitat where it's rocky and stuff is where we'd want to think about sampling, and also thinking about transported materials, probably a couple 100 meters down from those sites to make sure that we're collecting what's coming off of there. But we won't be going like hundreds of miles into the rivers. It will be, as Brittany said, like up from the mouth to make sure we're not getting the lake waters, but it will be, I guess, near lake, if that's even a term.
That's a good term. Cool. Amanda, do you have any questions about anything if you're still on the line? No.
So you've got several months that you're doing this. You're doing one sample per location. Are you trying to get more than one sampling on each tributary?
Yeah, so we're hoping to, if you look at the map on the screen, we have some priority sites where there's some known historical use by the Coregonids. So we're hoping to sample those locations multiple times, hopefully up to 6 or 8 times. And then also those yellow ones, the yellow stars on the screen, we are hoping to at least get one sample at those locations, or well, one collection at those locations, which might be more than one sample. So like I think 2 samples at those spots.
And we're also going to be using an underwater, like remote underwater vehicle that has a camera. So we're gonna, before sampling at these sites, do some site recon to make sure that we can find the habitat, see what it looks like. And if we do get a hit, so let's say one of these sites, there was a detection of something, we can also send it down to just get some eyes on it. Can we see it? Help us verify that. So I think that's a cool supplement to the eDNA work too.
I'm assuming you don't need fly fishers with waders to go in and collect actual samples of the fish. Darn it.
I mean, if you're volunteering for that, Frank, I'm sure we can find something for you.
Sounds good. Sign me up. Sign me up.
Awesome. Yeah. Amanda, you can stay in contact with me to volunteer, or I can or Mary would be your contact for that. Yeah. Cool. All right. Well, Frank, Janet, and Amanda, it's just you three left. You guys have any other questions or anything, and we can close out for the day?
No, you did a very good job. Very interesting. Very informative.
Thank you, Frank.
Just keep us posted, please.
Mhm.
Of course. We're very excited to have you all join us. So thank you so much for joining, and thanks for Megan.
Yeah, there'll be a follow-up email sent out to everybody as well with resources for you. in the recording.
Super. Thank you.
You're welcome. Thank you.
Lori, well, I
Good
hope you all
job.
have a fantastic Wednesday. Get outside and enjoy our almost summer weather.
We'll get there.
Yeah. All right, everyone. Have a great day.
You too.
Bye, everyone.
See you later. Thanks again.
Bye.
Great job.
Cool. Great job, guys. That went well.
Yeah, I agree. Oh, wait, let me stop. Hang on.